today's episode of Myeloma Crowd Radio, a show that connects patients with myeloma researchers. I'm your host, Jenny Elstrom. We'd like to thank today's episode sponsor, Amgen, for their support of Myeloma Crowd Radio and myeloma patients. Now, before we get started, as many of you know, last January we invited a prestigious scientific advisory board of myeloma experts to join us to help find exciting research being done for high-risk myeloma patients. We also invited a team of educated myeloma patients to participate. The goal was to serve an unmet need, essentially, for high-risk patients by raising funds for projects that would not otherwise get funded that were critical for patients who today are not having great outcomes. So we began that process last January. We submitted research proposal requests to hundreds of myeloma researchers, and we received back 36 very high-quality proposals. Those were vetted down to a list of 10. We did a radio show on each of the 10, and then we selected 2. So we have been fundraising since the fall for these specific projects, including, as many of you know, our 12-day challenge, and we have successfully now raised over $150,000 so far, with a goal to hit $200,000 by year's end. We had a very generous anonymous donor just donate $30,000. So as a shout out, I just want to say an incredible thank you to that donor and all who have participated in this process. It's becoming a reality, and these doctors will be able to get started on the research they're doing beginning in January. So this is the first time that a united group of patients, including all of you, have joined together to help fund the road towards a cure. The field is looking so very hopeful, and we're just thrilled with all of the new developments. Now, speaking of new developments, Dr. Robert Orlowski from MD Anderson Cancer Center is joining us today to talk about all these new advances and recent announcements, especially coming out of ASH. So, Dr. Orlowski, welcome to the program.
Well, Jenny, thanks very much for having me, and I wanted to take the opportunity to wish everyone out there happy holidays and best wishes as well for the new year.
Oh, thank you so much. Now, let me introduce you first before we get started, and then we have quite a bit to talk about. Dr. Orlowski is Professor of Medicine in the Department of Lymphoma and Myeloma with a Florence Maud Thomas Cancer Research Professorship in the same division. He has a dual appointment and is also Professor in the Department of Experimental Therapeutics. Dr. Orlowski is Director of the Myeloma Section in the Department of Lymphoma and Myeloma at MD Anderson. He's a member of the NCI Steering Committee, the Multiple Myeloma Tissue Bank Steering Committee, multiple committees, and the American Society for Biochemistry and Molecular Biology. He's the chair of SWOG, and if you're not familiar with that, it is a Southwest oncology group which helps create and craft clinical trials for multiple myeloma. And he is also on the editorial board of Hematology and the Journal of Clinical Oncology. He's received numerous awards over a number of years, including the LLS Scholar in Clinical Research, the LLS Man of the Year, the Emil Frey III Award for the Excellence in Translational Research from MD Anderson, and he's the recipient of a very coveted SPORE grant from the NIH, which is awarded to top researchers and is quite a rare award. So, Dr. Orlowski, thank you so much for joining us. I think you had in mind maybe giving us a top 10 or top 12 kind of abstracts on ASH or topics on ASH that were covered. Just so if patients don't know what ASH is, do you want to just begin by telling them what that is first?
Sure. Well, ASH stands for the American Society of Hematology, and once a year, usually early in December, there's an annual meeting, and this is where researchers from all over the world get together to present their most exciting and most recent data. And myeloma, of course, is one of the hottest diseases out there in terms of new drug development. So it's often an exciting time when we get to see what people are doing all over the world to combat this disease. What I'll do is give you a quick overview of what I thought were some of the top abstracts. And, of course, there are many wonderful presentations. and we don't have time to cover them all, but I'll do my best to hit what I think are the highlights. First of all, you mentioned earlier about SWOG, and I think one of the most important presentations was a SWOG-led study, which was for newly diagnosed patients with myeloma, and they were randomized to receive either lenalidomide and dexamethasone or len and dex, but with the addition of bortezomib, and the three drugs showed a better response rate, a longer time in remission, and also a longer overall survival. And this study is important because for many years we've been using this so-called RVD regimen as the sort of de facto standard of care, but this is really the first study which shows that, in fact, this combination is a superior one to a two-drug combo. A second abstract that I thought was very important was a study from the French myeloma group predominantly, and what they did is they compared bortezomib, cyclophosphamide, and dexamethasone, which is also known as Cybor-D, to bortezomib, thalidomide, and dexamethasone as an initial therapy prior to stem cell transplant. And for many years, Cybor-D has been talked about as being one of the best regimens, and the same has been true for bortezomib, thalidomide, and dexamethasone. But it's always helpful to have a comparative study, and this trial did exactly that. And interestingly, what it showed is that bortezomib with thalidomide and dexamethasone looked better. So Cybor-D is certainly still a good option, but I would probably, if I had the choice just between these two, go with bortezomib and thalidomide and dex, or as per the prior presentation, bortezomib with lenalidomide and dexamethasone. A third study to highlight was an exciting one that had been done by the French, and we actually are also participating, we being many centers in the United States led by the Dana-Farber, as well as with MD Anderson. And here in this study, patients are getting bortezomib, lenalidomide, and dexamethasone. Then they get randomized. All patients have their stem cells collected. Half of the patients get an early transplant. The other half get more chemotherapy and don't go on to transplant until there is evidence of progression of their myeloma. And the results showed two interesting findings. One endpoint was what's called progression-free survival, and that means time until the myeloma begins to progress. And as you would maybe expect, the people who got the early transplant with the high-dose melphalan had a longer PFS than the patients who got only chemotherapy. However, when you look at the overall survival, because the patients who got only chemotherapy, even if they progressed earlier, were able to get a transplant at that time, the overall survival for the two groups was essentially identical. And that suggests the possibility that at least for some patients, it may be possible to delay stem cell transplant, especially if they do very well with their initial chemotherapy. And we'll probably have some questions about that a little bit later on that we can get to. The next abstract that I wanted to cover was a study that we actually did at MD Anderson. We know that lenalidomide, bortezomib, and dex is a great regimen, but it still doesn't achieve complete remission in all newly diagnosed patients. So what we did is we added panobinostat as a fourth drug. Probably many of you know that panobinostat is approved for relapsed myeloma, and typically if a drug works there and you give it to newly diagnosed patients, it works even better. And in this update of our study, we found that the four drugs were tolerable and that there was an indication that maybe they worked better than just the 3-drug RVD combo. And actually about half of patients were able to achieve MRD negativity even before they went on to stem cell transplant. So that's exciting because if you're MRD negative before the transplant, you certainly will probably be after the transplant, and your outcome after the transplant should be better as well. One other thing to mention in the abstract category for recently diagnosed patients, and this has some application to the high-risk setting you mentioned, Jenny, because in addition to the great initiative that you've put together to tackle high-risk myeloma, we also here at MD Anderson have been interested in that. And so we have what's called a moonshot for high-risk myeloma, which was basically a donor-funded effort with a number of different approaches. And the one that was presented at ASH as an oral presentation was the use of natural killer cells, or NK cells. These are cells that are part of the immune system and that can have anti-myeloma activity. And what we did in this trial is that patients with high-risk disease who were getting stem cell transplant, right after the high-dose melphalan, which is the main chemotherapy with transplant, they were getting, in addition, cord blood-derived, laboratory-expanded NK cells. And the thought is that by doing so, you will have not just the benefit of the melphalan chemotherapy, but you'll have an immune attack against the myeloma with the NK cells. What we found is that the NK cells were very well tolerated. There were virtually no side effects. And although the follow-up is still short, there was an indication that the response quality looked very good. And so this was something we were excited about, as were the reviewers, and we hope to be able to expand upon this further in the future and treat a larger group of patients.
And
so those were abstract.
Well, before we go on, do you want to describe the Moonshots program at MD Anderson? Because I'm familiar with it, but I don't think most people are.
Oh, sure. Well, thanks for asking and for the opportunity. One of the things that our president at MD Anderson, Dr. DePino, did is he wanted to, of course, as do we all, but he wanted to focus on making a big difference over the next 5 years in certain cancer areas. So through the generosity of many donors, he was able to raise a war chest, if you will, of funds, and then he put together a group of cancer experts to review applications, and he sought proposals from all over the cancer spectrum, not just myeloma, and those proposals which were felt to have the best chance of making a big impact were selected for funding. And one of those which was selected was this high-risk myeloma effort. And what we're trying to do now is expand further beyond the study that I just mentioned to you, and also develop chemotherapy drugs as well as immune therapies that hopefully will be specially targeted against high-risk myeloma. And the exciting thing is that many of these will also be applicable to standard-risk myeloma, but we're testing them first in high-risk disease because, unfortunately, patients with high-risk myeloma relapse sooner. The only good thing about that is that if we try a new treatment and it works, we will know more quickly if it works in that setting than in patients with standard-risk disease.
Makes sense.
So the previous abstracts that I reviewed for you briefly were ones that dealt with recently diagnosed myeloma, and now I'm going to move into some of the things in the relapsed setting. So abstract number 6 was the presentation of a study with an oral proteasome inhibitor. The generic name is ixazomib, and this was a large phase 3 trial which compared lenalidomide and dexamethasone to ixazomib with lenalidomide and dexamethasone. And the study showed about a 6-month improvement with a longer time in remission, and the tolerability of this combination was very good as well. And you probably know that this drug was approved by the FDA in November because of these data, and I think it's a big improvement from a convenience perspective, because instead of having to be injected with either bortezomib or carfilzomib, in some cases you may now be able to take this medication orally. Another abstract for relapsed myeloma was an update of data from the elotuzumab study. Elo is a monoclonal antibody which binds to a protein on myeloma cells and makes them more visible to the patient's own immune system. And as you probably know, this was also approved in November in combination with lenalidomide and dexamethasone. And this abstract that I'm referring to gave longer follow-up, now 3-year safety and efficacy data, and it showed a continued benefit for the group of patients who got the Elo-Len-Dex. And I think that's exciting because in some studies we see an early benefit, but that may not always be maintained later. And in this trial, it actually continues to show a difference. Elo wasn't the only antibody around which there was excitement in myeloma at ASH. And the next abstract looked at a drug called pembrolizumab in combination with lenalidomide and dexamethasone. Now, Pembro is a drug that is already approved for some other cancers, and some of you may be aware of President Carter's melanoma, which is being treated with a drug that has the same mechanism of action. And we're now trying this in myeloma, and this presentation showed that the drug looked active in the combination, and there was some excitement as well because even patients whose myeloma had previously grown on lenalidomide and dexamethasone seemed to respond when this antibody was added. And this will be another immune therapy that maybe could be combined with other antibodies in the future. So that's exciting anytime.
Is that a monoclonal antibody, or that's a checkpoint inhibitor, or what is that?
Well, good question. In this case, it's both. This is an antibody which works by blocking some of the checkpoints. So checkpoints in immune function have to do with the fact that cancer cells often suppress the patient's own immune system. They do this to survive, and we now understand some of the mechanisms which are used by the cancer cells, and these checkpoint inhibitors take that inhibition away. So if you will, they boost the patient's immune system and therefore provide additional benefit by allowing it to attack cancer in general, and in this case it was multiple myeloma.
That's great.
Now, a couple other abstracts to mention quickly. Number 9 was a study that looked at a different oral proteasome inhibitor called aprozimib. I told you earlier about ixazomib, but there's another oral drug in this class called aprozimib, and it was combined with pomalidomide and dexamethasone, and it showed a 50 to 70% response rate, which is pretty high in the relapsed setting, and I think we're looking forward to additional studies of that drug in the future. And getting to the finish line here, abstract number 11 was a study of a anti-CD38 antibody called isatuximab. Many of you are probably familiar with daratumumab, which is a drug that's been FDA approved and also binds CD38. But there's another antibody against the same target, and an update of the data at ASH showed that it works quite well, and because it binds to a different portion of the target than daratumumab, it's possible that, in fact, the two could be used either together or sequentially. And then abstract number 12 on my list was a late-breaking abstract which had to do with chimeric antigen receptor T cells. Probably you've heard about these. There's a lot of excitement about them. This is a technology where you can take out the patient's own T cells, modify them in the laboratory so that they can better recognize myeloma cells, and then infuse them back in so that they can kill the patient's cancer. And this was a study from the National Cancer Institute which showed that even in very heavily pretreated patients, responses could be seen. This was a still relatively early trial with only a handful of patients treated, but really was exciting because of the fact that this technology, I think, is going to really be very more broadly applicable to myeloma patients. And then finally, I'll give you a baker's dozen. So abstract number 13, we've mostly talked about myeloma right now, but there's a related disorder called light chain amyloidosis. Some patients have this by itself. Other people have both myeloma and amyloid. And amyloid is a disease where the monoclonal protein made by myeloma cells can clump together and be deposited in various tissues and cause damage to those tissues. And the same chemotherapies can be helpful as in myeloma against amyloid, but what they tend to do is they kill the cells that make the protein, but they don't get rid of the clumps of garbage protein that have already been put down. But there was a really exciting abstract for amyloid with a new antibody called 11-1F4, which actually looks like it's able to bind specifically to the amyloid fibers and make it easier for patients to get rid of that junk protein. So very exciting, could really change the landscape for amyloidosis. So those were my top 12 plus a baker's dozen of things that I found exciting from ASH.
Well, that's perfect. There's so much content in what you just said. I'm not quite sure what we should talk about first, but maybe we talk about the monoclonal antibodies, and then as we hit these topics, we'll go back to these top 13 and ask some follow-up questions. So sure. Daratumumab was approved. Elotuzumab was already approved earlier, right? And,
correct.
And so I went to a session where Dr. Jagannath was talking about how to use these and when to use them. Do you want to cover how does a doctor decide when to use these? And when would you add them? When would you, it depends on how they've been approved in part, I guess. But as a myeloma specialist, what was your perspective and takeaway after hearing about all of that?
Yeah, that's a great question. The exciting thing is that elotuzumab and daratumumab, although they're both antibodies, they bind to different targets on the myeloma cell, and so that raises the possibility that they could both be used, albeit at different times. As you pointed out, right now they've been approved in different settings. So elotuzumab was approved with lenalidomide and dexamethasone in patients with 1 to 3 prior therapies, and so that's the setting where we would most often at this point use it. Daratumumab was approved right now by itself, and for people with 3 or more prior therapies is one of the common ways that it's given. So probably at least initially we would tend to use the ELO with Len-Dex earlier and the daratumumab later. But sometime next year, we're going to get data of daratumumab with lenalidomide and dexamethasone versus Len-Dex in the same patient population for which the ELO with Len-Dex has been approved. And although it's not always fair to compare two different studies, we'll at least be able to get some idea of whether one looks better than another, although the best way really would be to do a head-to-head comparison.
Mm-hmm. Well, I think that'd be really helpful for patients. So when you say 3 or more prior therapies, if you've gone through transplant, your induction therapy and then transplant, you're going to be at least to 3, right? So let's say somebody relapses after transplant, which do you choose at that point? Like ELO with Len-Dex or daratumumab with another combination, or what do you do? I guess it just depends on the patient, but...
Well, it's very much dependent on the patient. There's no one-size-fits-all. But the way that we define what's called a line of therapy is that it's a planned treatment which sometimes can include many different chemotherapies as long as the myeloma does not progress. So, for example, if you're newly diagnosed and then you get bortezomib, lenalidomide, and dexamethasone as initial therapy, and then you get transplant, and then let's say you do bortezomib as a maintenance therapy, as long as the myeloma hasn't progressed at any time during that period, that's considered one line of treatment. And so when the myeloma does progress, you're actually only in your second line, even though you may have gotten 4, 5, or even 6 drugs.
Okay, I didn't know that. I thought it was just treatments because, that's very interesting. Okay, well, that makes sense. And let me go back to my notes here. The isatuximab is the same. Is this the SAR drug by Sanofi?
It is, correct.
Okay. So that's the one that is usually SAR and then a lot of numbers after it. I don't really ever remember what that is, but now it has a name. And so you're saying you could potentially use that with daratumumab. So that's very fascinating because they're hitting the same target.
Well, that's a theoretical possibility. So far, they have not been used together. And the first thing that we really need to do is to try to get isatuximab approved both alone and in combination with lenalidomide and dexamethasone. But yes, because they attach to a different portion of CD38, which is the target, theoretically one might be able to use them together, although that doesn't necessarily mean that the combination would be better than either drug by itself.
Mm-hmm. And I know we're getting addicted to having drugs be approved pretty quickly as myeloma patients, or it's very exciting to see what happened this year especially. So when do you think that drug will be FDA approved? Because it seemed to be behind the others, but is up and coming too.
Sure. Well, there were 5 approvals in 2015 and 3 in November alone. So 2015 is going to be a tough year to beat.
Yeah.
I think the isatuximab probably would be either late 2016 or more likely sometime in 2017. And then other drugs that look exciting that could get an approval sometime in the next couple of years would include drugs like selinexor, which is a drug that blocks proteins moving from one part of the myeloma cell to another. And I do think in the next couple of years there's a possibility that we will have even one of the CAR T-cell therapies approved for myeloma. And there are some other drugs that look quite exciting as well. So I think it's a very exciting time in terms of getting new therapies available. And with every new treatment which prolongs the ability to keep people in remission, I think we come closer to a cure.
Yeah, I believe so too. It's just really exciting what's happening. Well, let's talk about, secondly, you mentioned ixazomib, and sometimes people want to know, is this just an oral version of Velcade? So can I call my doctor tomorrow and say, could you swap me off, Velcade and put me on ixazomib? As a myeloma specialist, what would you say? Because I'm sure you're going to get those types of questions.
Sure, and that's certainly an exciting option. The data that we have so far with ixazomib, for example, from the trial that I described earlier, the ixazomib was taken right from the beginning. So people did not start on bortezomib and switch over, and we don't really have a lot of data about that approach yet. So what I would tell patients is that if they're on bortezomib and they're doing well in terms of side effects, and the myeloma is responding well, I would probably stick with that. If they're on bortezomib and having lots of problems like neuropathy, then I think it's reasonable to look at switching to ixazomib because the drug does have a much, much lower risk of neuropathy than does bortezomib. So for right now, that's the one setting where I would consider switching.
And it seemed to be that they were having pretty good results with the ixazomib, even for high-risk myeloma. And I know bortezomib has good impact, like in deletion 17 patients. But did you have any indication for high risk about what that drug might do?
Well, one of the groups that was followed very closely on that trial were patients with deletion 17p, which is one of the high-risk features, and the study did show that addition of ixazomib made a significant difference with an improvement in their outcome. So I do think that patients with high risk should definitely be considered for a proteasome inhibitor of one kind or another, which could be bortezomib or carfilzomib or ixazomib. But even with those drugs, high-risk patients still don't do quite as well as the standard-risk patients, so we're going to need to get additional drugs approved to make that difference be hopefully smaller.
Okay, perfect. And I know there was a study or a paper on a study that was done on Kyprolis or carfilzomib versus bortezomib or Velcade. Do you want to comment on that at all? I didn't attend that session, but I would have liked to.
Sure, yeah, and you're right. That was a very exciting presentation. What was done in that study is that bortezomib and dexamethasone was compared to high-dose carfilzomib and dexamethasone for 1 to 3 prior therapies. Now, the difference between standard dose carfilzomib and high dose is that the currently approved dose, patients get 20 milligram per meter squared, and then it goes up to 27 milligram per meter squared, but no higher. In this study, it was first 20 and then 56 milligrams per meter squared. And it did look like the time in remission for the carfilzomib with the high dose and then also dexamethasone was about twice as long as it was for bortezomib and dexamethasone. And so probably, although this won't be necessarily a new drug approval, but the current approval for carfilzomib will probably be modified to indicate that a higher dose may have an increased benefit. And I say may just because in the trial that we're talking about, there wasn't a head-to-head comparison of the standard carfilzomib dose versus the high dose. We're actually doing that as part of a SWOG trial. So we hope to have some of those results presented at next year's ASH meeting, which will be in San Diego.
Mm-hmm. Yeah, that will be a very nice one to attend. So is that's my question, I guess. You kind of made that point. Is it fair to compare a higher dose of one to a lower dose of another, or does that mean you might want to increase the dose with all the proteasome inhibitors? I mean, you might not want to if you have neuropathy issues, but if you have
Well,
oral versions that don't have as much neuropathy, and maybe you can bump it up. I don't know.
Well, it's a very good question, and one thing that we know about new drugs when they get approved is that we learn more with each year about what the best dose and schedule of these drugs could be. So the chances are 2 or 3 years from now, we'll probably be using some of these drugs in a different way than we do now. For example, with carfilzomib, right now, the most common way to do it is to do 2 days in a row IV for 3 weeks on and 1 week off. But there are several studies that are looking at the possibility of doing the carfilzomib at an even higher dose than the 56, but just 1 day per week for 3 weeks on and 1 week off. And that would be exciting because if you can get the same benefit, or who knows, maybe even more, but not have to come in for 3 infusions because instead of 6 you're only doing 3, that would be a win-win for everybody.
Oh, yeah. Less time in the clinic is better for everyone. And the other proteasome inhibitor, the other oral one that you mentioned, is that taking carfilzomib, that proteasome inhibitor, and kind of turning it into an oral version as well?
It's not exactly the same as carfilzomib, but the chemistry is similar, you're right, and we think that sometime in 2016 there will be larger studies with that drug available if people are looking for new treatments, and we hope that maybe a few couple years down the road from that, if the trials are positive, there would be an approval, and then we would have 2 oral proteasome inhibitors to pick from.
Mm-hmm. Yeah, that's a challenge. I mean, it's a blessing, a great blessing, but it's also a challenge, and we were talking about this before the show started, and it's one of our write-in questions that I probably should say for the end, but I'm going to ask it now. So this seems to be pretty complicated for the myeloma specialist to pick and choose and know what to use when. How do you help the local oncologist who's treating patients and they didn't attend ASH and they don't know all the detail behind it? What do they do?
Well, it's important, of course, to try to stay current with all of these drugs, and I'm always impressed at the knowledge of the local oncologists, quite frankly, because me being at MD Anderson, I get to focus just on myeloma, and yet they have to keep up with every drug in every type of cancer. And yet when I talk with them at meetings, they seem very up to date about what's going on. I do think, however, that especially given the number of new drugs approved, one thing to always suggest is that it's never a bad idea to consult with a myeloma expert so that you have an opinion from somebody who specializes in that cancer. In terms of giving advice on which of these treatments should be used, that's a little bit tough in a kind of generic fashion, because I think that what we need to try to do is understand better whether certain therapies work best for some patients, whereas others work better for a different group, and we're not quite there yet. But as of right now, I think in the relapsed setting, the most exciting combinations really look like carfilzomib with lenalidomide and dexamethasone, and also potentially elotuzumab with lenalidomide and dexamethasone. So those 3 drug regimens look really great. And then also from a study that we led at MD Anderson, carfilzomib with pomalidomide and dexamethasone looks really exciting. So if I were relapsing after my initial therapy, those would be the 3 things that I think would be most likely to result in a great benefit.
As you said at the beginning of the show, that it's becoming more clear that these triplets are better than these 2, doublet drugs. I'm just looking at that list, carfilzomib, Lendex, and Elo Dex, Lendex, and Carpalm Dex. I'm wondering if someday you could take that Dex out of that combo, but maybe that's just wishful thinking.
Well, it's a great question that lots of people ask because the Dex is nobody's favorite drug, that's for sure.
No, everyone hates it. Yeah. Well,
part of the problem is that it really does seem to add to the benefit of almost all of the chemotherapies that we give, but where things may change is with these immune treatments like the antibodies that we mentioned. Many people are concerned that steroids, which of course suppress the immune system, may actually be holding back the efficacy of some of the drugs we've talked about, like the ELO, like the Dara, like the Pembro, and maybe the same thing will be true for the CAR T cells. So I think there's a lot of interest, not just on the patient side, but also on the doctor side, to look at ways that we could at least reduce the steroid dosing. And I do think eventually we'll probably be able to get rid of them, but we're not quite there yet.
Mm-hmm. Okay, well, I want to cover two topics, and we already have caller questions, so I will get to those, and we will probably go over. So don't be nervous that I'm not going to get your caller questions. But let's talk about transplant a little bit. So I attended a session by Dr. Geralt, who is at Memorial Sloan Kettering, and he was saying, people ask him all the time, with these new combination drugs that are coming up, can we, ultimately get rid of transplant? Because everybody would love to. But he said the role of transplant is still, melphalan is probably still the most effective single agent. And for people who just don't like transplant and don't do it just because they don't like it, he just said the data just doesn't support that approach. So what is your opinion on transplant as a whole? And then I thought that's interesting what you said about the IFM study about transplant, but about early versus late. You're saying it might not matter when you get that.
Well, I think that it's not so much early versus late, because the transplant that was done in the group that got a delayed transplant, that was part of the second line of treatment. So I do have some patients who come and say, listen, I want to leave transplant for the absolute last thing that I do against myeloma. And I tell them that I think that's a mistake, because if you have myeloma that has relapsed after 5, 6, 10 prior lines of therapy, the transplant is going to be much, much less effective. But I do think that the data showing that transplant as part of the second therapy is roughly equal to transplant given as first therapy are becoming more and more robust. So what that says is that it's possible. Now, remember, this is a hypothesis. We haven't proven this. But it is possible that if you're newly diagnosed and you get into a complete remission with just chemo alone, and especially if you have MRD negativity, it may be okay in that setting to collect stem cells and store them away, and then just do a little bit of maintenance therapy and not do a transplant up front. We really need to do a study like that. But if you don't achieve a complete remission, and if you're not MRD negative, then doing a transplant to get to that point, I think makes a lot of sense.
Well, that makes a lot of sense, and so that provides a lot of clarity around that, because I do have friends that have, tried to do just different combination therapies that weren't really working, and then the transplant, when they try to do it, it doesn't work. So
yeah,
that's
unfortunately, myeloma still has a tendency each time it relapses to be more resistant to treatment. And so you want to try to do the transplant early rather than as your last line of treatment.
And, yeah, it's kind of confusing for patients because they don't really know what early means sometimes, They don't know if that means, oh, gosh, within a month of being diagnosed, I need to be getting a transplant. So that's very informative. Thank you for helping clear that up.
Sure.
The last thing I'd like to talk about before we go to some questions, and we can talk about this for a while, is the whole field of immunotherapy. And maybe we talk about it first as it relates to transplant, because what I see people trying is, let's go to transplant first, kind of empty the cup, as some people talk about it, of the tumor burden, unload the bulk of the tumor burden through transplant, and then try these immunotherapies both as your immune system's ramping back up in a normal way after transplant. And so maybe you want to talk about how transplant would be used with immunotherapies, and then maybe how it would be used without immunotherapies.
Sure. Well, that could be probably a topic for a whole 1 hour show just
probably.
by itself. There are, of course, different immune therapies, and some of them seem to work better if there is less disease. For example, vaccines. Probably, at least with the technology that we have right now, vaccines are probably going to be challenging to get to work if you have lots and lots of any cancer. But if you have only a small amount, then vaccines might be a good way to build up immunity and get rid of what could be left over. If you have more cancer, then I think some of the therapies we've talked about, like the antibodies, the checkpoint inhibitors, and the CAR T cells, are fairly reasonable ways to go. The advantage of the transplant is that with the high-dose melphalan, you're able to kill off quite a few of any leftover myeloma cells, and because you're putting the patient's stem cells back in, you're able to hopefully restore their immune system to a point where it may be better able to attack the myeloma. Now, of course, if you do an allogeneic transplant where you have stem cells from a different donor, you don't donate to yourself, an allotransplant has always been said to potentially be curative. The problem there is that the toxicity of that approach is much greater. So I think that what we're hoping to do in the future is to either find some combination where we do a transplant and immunotherapy, or what we're actually going to be doing sometime next year at MD Anderson is we're going to have a trial where newly diagnosed patients are actually going to be started first on immunotherapy for a period of time, and if their myeloma responds, they're going to continue on that. If it doesn't, then we'll put them on standard chemotherapy. But we're going to actually try to find a way to get rid of chemotherapy and replace it with immunotherapy completely.
Mm-hmm. And which immunotherapy would you be looking at to be the most powerful one to start in a newly diagnosed setting like that? The CAR T cell?
Well, we have a number of combinations that we're going to look at, and each of the patients will be studied closely to look at the effects on the immune system that these treatments have, because right now we have a couple of antibodies that are going to be part of this, but we don't know for sure if these are the best ones, and we're hoping, therefore, to be able to build on that as a platform for the future.
And I know that some facilities are starting to run, like the NIH, the one that you mentioned, they were targeting BCMA. And I heard someone that attended that session say that the next time they do it, they're going to have patients who have a lower tumor burden, like they have to have 50% or less tumor burden, so they don't get the cytokine release syndrome. So it sounds like they're learning as they go how to make it, number one, more effective and then more safe. So I guess my question is, because these CAR T-cell therapies are therapies and not necessarily a drug, how long does that process take to get that through the clinical trials and kind of into the clinic? Because sometimes with a drug it can take a very long time. Is it faster to do these immunotherapies, or because it's not necessarily a drug, it's more like a manufacturing process?
Well, it depends a little bit on the type of immunotherapy. If you're talking about just an antibody, even though that's sort of technically an immunotherapy, it's still similar to a drug, so those tend to be a little bit faster. The CAR T cells, because they involve most often taking out some cells from the patient, manipulating them in the lab, and then putting them back in the patient, those often take a little bit longer to get going because the process is more complicated to develop the treatment, and therefore to make sure that the safety factor is maintained, it takes more time to prove that you can do it safely. But what we also are working on here at MD Anderson, and we're not the only place, but I think we're one of the major places this is happening, is we're working on using these chimeric antigen receptor T cells not from a patient, but from a different donor. The advantage of that is that you don't have to wait a long time to generate each cell individually from each patient.
Oh, okay. It looks like Dr. Orlowski has dropped for a minute, so let me just call him back because he was in the middle of saying something very, very important.
Dr. Orlowski's office.
Okay, this is Jenny Ahlstrom. We're on Myeloma Crowd Radio. Can you patch me in because his call dropped off the show?
Yes, one moment.
Thank you.
Hello?
Okay, Dr. Orlowski, sorry, I don't know why that dropped, but you were in the middle of saying something that we all wanted to hear. So you were basically
sorry,
saying,
where did I get dropped off?
So you were saying that instead of having the CAR T cells come from a specific patient, they were coming from a general donor, you were talking about the advantage of doing that you don't have to wait,
great.
and yeah.
Well, I apologize about the drop-off.
Oh, no,
I think maybe our
sister.
time had expired and they decided to cut us off, but I'm glad you were able to get me back on. So the problem with the auto CAR T cell is that we have to take cells from each individual patient. We have to then modify them in a lab. We have to grow them up so that there's a certain number and then infuse them, and that takes time. If you can take the cells from a donor, then you can save time because you can use the same cells for several different patients, and also it makes it a little bit cheaper, although that's not the major motivation, I would say. But if you do this allogeneic donor, then it should be much faster to get to each patient individually.
Well, I guess you could pre-manufacture them at that point, right, and have them be ready to go, or is this... So our companies are starting to look at this manufacturing process and trying to figure out how they jump in the game on this, or facilities are doing this?
Well, that's actually a combination of both, because oftentimes what happens is that we work with companies to generate the cells, and then when they're shipped, they go through what's called the GMP lab here, or the Good Manufacturing Practice Laboratory, to make sure that the cells are safe and they're not contaminated. So those sometimes the same laboratory as is used for stem cell transplant.
And would they be as effective, or would they be more effective coming from a different donor?
I don't think we know yet for sure. The technology is similar, so there should be equal efficacy. I think the only concern about an allogeneic or a donor cell is the possibility of the graft versus host reaction, as there is with an allogeneic transplant. But we now have some tools to try to modify the donor cells so that hopefully that reaction will not occur.
Okay. All right. Well, we will look forward to seeing what everyone, including you, are going to do on that. That's very, very exciting. And that's actually one of the studies that the MCRI is funding, is a CAR T-cell project for BCMA and CS1. So it's very exciting to see what's happening. So you touched on this a little bit. This is my last question, then I'll open it up for questions, and if we have time, I'll ask a few more. But you touched on the checkpoint inhibitors, and so I read the paper on it, and the results were looking good, but you had mentioned another one that had like 50 to 70% overall response rate, and it didn't seem like they were that high, but everyone, I saw a lot of activity around that, or a lot of excitement around that. So what am I missing about that makes these checkpoint inhibitors so exciting as kind of one of the next big things to watch, or is it?
Well, I think the excitement about that presentation was not so much the overall response rate, but the fact that there were patients on that study whose myeloma had grown through lenalidomide and dexamethasone and then responded just with the addition of this checkpoint inhibitor. And so that makes it exciting to think about the response rate if you gave the 3 drugs right up front to someone who hadn't been exposed to any treatment at all.
Oh, that makes sense. You don't have to worry about them relapsing, and you just use it right up front.
Correct.
Okay, that's fascinating. There's so much to talk about. Well, let me open it up for caller questions. If you have a question for Dr. Orlowski, you can call 347-637-2631 and press 1 on your keypad. So we'll choose caller at 371-3695. Go ahead with your question. Hello?
Yes,
caller.
hello?
Go ahead with your question.
Hey, Dr. Orlowski, how are you?
Good. How are you?
Good. I really enjoyed your presentation. The thing that I have 2 questions about Revlimid and the salvage studies. Particularly the the one with Elo. Are these patients naive to Revlimid, and where do you find them? Or where did Dr. Loniel find them? And then
I'm sorry.
No,
Go
that's
ahead.
it. Great. And then the second question is, there are a lot of people who've been maintained on very low dose Revlimid, let's say 5 milligrams, and have progressed. It sort of ties in with the first question. And would you then, give them Elo RD at the higher dose of Revlimid? Or would you just assume that that's a waste of time, considering they've progressed on a smaller dose of Revlimid?
Yeah, thanks very much. It's a very good question. The studies that led to the approval of Elo, and indeed many of these phase 3 trials that lead to approval, are done not just within the United States, but are done internationally. And practice patterns are different in different in different countries. So in some areas, it is easier to find patients who have not been exposed to lenalidomide before, or who were exposed but were sensitive and then had myeloma relapse off of treatment. So for the elotuzumab study, you could not have lenalidomide refractory disease. What that means is that you could not have myeloma progressing on lenalidomide, even a low dose, to be able to get on that study. So the answer to your question is that we don't know how well Elo-Len-Dex would work in somebody whose myeloma is progressing on lenalidomide at, say, 5 or 10 milligrams. I would think it's still worth trying, and my prediction would be that the combination would work, but probably not as well as in people whose disease is not progressing on lenalidomide.
Thank you very much.
My pleasure.
Okay, great. Thanks for your question. We have another caller at 983-6757. Go ahead with your question.
Happy holidays, Dr. Orlowski and Jenny. This is Dana Holmes. Thanks so much for taking my call. Dr. Orlowski, I realize that the smoldering abstracts didn't make the top 11, but I have a two-part question and follow-up related to smoldering myeloma. So if you would just bear with me. My first question is, if you could select one and only one of the available or soon-to-be available early treatment intervention clinical trials for a high-risk to progress smoldering patient with a standard risk disease profile, for instance, 11-14 translocation, what would it be and why? And then the follow-up question is, if you could design a dream high-risk smoldering trial, if you may, using either the available drugs or those still being tested, what would it be and why?
Well, thanks very much, Dana, and I know we're followers of each other on Twitter, so keep up the good
we are.
work.
Always love your posts.
So you're right, the smoldering space, although there were some abstracts at ASH, I don't think any of them yet are what I would call practice-changing, which is why I didn't highlight any of them. Just to be a little bit on the advertising side, there is a national trial for smoldering myeloma, which is comparing lenalidomide to observation, so that's one option to consider. In terms of what drug classes I think are also exciting, probably the antibodies are ones that I would really think of. There was a study with elotuzumab, which finished enrollment. There is a study with daratumumab, and then early next year here at MD Anderson, we're going to open a study with isatuximab. So I think these are exciting because
Ixatuximab in smoldering myeloma?
Correct.
Oh, okay.
So the reason these are exciting is that, first of all, although the antibodies are not that convenient because it is IV and it takes a few hours to infuse, the side effect profile is relatively benign compared to some of the other chemotherapies because these are very targeted agents. If you take something like daratumumab or isatuximab, both of which have about a 30% response rate in very, very heavily pretreated patients, you would think that if you give it to previously untreated folk, the response rate should be at least double, if not higher. And the other hope is that if patients do respond but then eventually progress, because the antibodies are different classes of drugs, the myeloma would hopefully still respond to things like bortezomib and carfilzomib and lenalidomide and pomalidomide, so there would not be any cross-resistance. So those would be probably where we would like to go and where we are going. And I think after the isatuximab study, we also would like to learn from those patients, see how the drug affects the myeloma and the immune system, and build on that to develop some kind of antibody cocktail, which would hopefully do even better than a single agent.
Mm-hmm. Now, Dr. Orlowski, these antibodies, do they have more or less like a half-life? In other words, if once you give it, obviously it's working in your body, but once you stop it, does it typically then stop working? So would you need to continue this therapy if a patient, let's say in a smoldering stage, did respond?
Well, the half-life of most of these antibodies tends to be on the order of 10 days to about 2 weeks. And for those of you that don't know what half-life means, that means the time that it takes for half of the drug to leave the patient's body. And it probably will be needed, at least in the short run, for patients to continue on dosing, albeit with probably some reduced frequency. But I do think, let's be really dreamy here, because you said fantasize about what the best studies and outcomes could be. Mm-hmm. It's certainly possible that if a patient achieves a complete remission and is MRD negative, that one could consider stopping the antibody and then just monitoring MRD and restarting if the MRD turns from negative to positive.
Mhm. Okay, lots of hope, and I know Gary Peterson made you part of his dream team, so this is why I presented it to you as a dream trial. So thank you for your response, and thank you for what you do for the myeloma community, particularly for being so willing and available on these social media sites for patients. You're always so very responsive, and have a wonderful holiday, both of you.
Thank
Take
you
care.
so much, Dana. Thanks for your questions. Always great questions. Okay, a caller at 647-7789. Go ahead with your questions.
Yeah, hi, Dr. Orlowski. Thank you very, very much for a very informative session. Going back to the early versus delayed transplant, if a patient currently on CARPOM-Dex is in VGPR, so the protein is detectable but too low to quantify, so almost asymptoting to a complete remission. Do you advise doing the stem cell transplant sort of earlier rather than waiting for relapse? And a related question, the U.S. part of that French trial is a little bit different. I understand the Revlimid is not stopped after 1 year. Do you expect a different outcome in the trial on the early versus delayed question?
Well, thanks very much. Let me tackle the second question first. You're right. The French study had a limited duration of maintenance, whereas on the American side, there is maintenance until progression. And it's tough to know whether the outcome will be similar. That's part of why the trial is being done. And by the way, we still need, I think, a couple hundred patients to finish enrollment onto that trial here in the U.S. So if any of you are newly diagnosed, do think about that. In terms of the first question, whether you should do a transplant if you're in VGPR after CAR and POM and DEX, it's a little tough to sort of give a blanket recommendation without knowing more detail. But in general, the higher the response quality, the longer the time in remission. So if the VGPR can't be turned into a CR with more chemotherapy, I think there's a strong rationale to think about adding the transplant at that point. But I would need to know a little bit more about the case to be sure.
Thank you.
Thank you.
Okay, great. Thank you for your questions. Now, Dr. Orlowski, I had a question for you. Just two quick questions. I saw in one of your articles that you posted in your paper, and maybe I should mention this right now because you have a paper, they're called Paperlies, which are online newspapers, and you have one where you pull different articles that are of interest to myeloma patients. And following that kind of gives you an idea of what's happening in myeloma. But in one of those articles, you said that they mentioned that there was a specific gene that might indicate whether somebody responds or not. So are we getting closer to understanding which drugs may be better for which individuals based on the genetics that they have?
We are. We're not quite yet at the point where we can do these genetic tests and determine exactly what drug or combination is best. But actually, if we go back to ASH, one of the interesting abstracts was about a panel of 14 genes whose expression could predict sensitivity or resistance to lenalidomide, and we're developing similar panels for bortezomib. And I think that hopefully in the future what will happen is that we'll be able to use these biomarkers to better predict what combination is best for each individual. And the advantage of that is that nothing right now gives 100% complete remission in everybody. And so we, to some extent, even with standard therapies, it's a trial and error. Let's try what we think is the best combination and see how it does. If we could predict ahead of time what combination is best, we would be able to get more people into complete remission, and we would do it more quickly because we wouldn't have to try something that wasn't going to work. And of course that would save money as well, because why give somebody a chemotherapy if their likelihood of a complete remission to that treatment is low? But we're not quite there yet. Give us a couple more years and I think we'll be there.
Well, that sounds really exciting. So I guess that follows into my last question is, what do you see as a pathway to a cure for myeloma? Because people are starting to say that really publicly for the first time.
Well, I think the immune therapies, the antibodies, and these CAR T cells are probably going to be the things that hopefully get us there. So those with the chemotherapies and the stem cell transplant put together is going to be probably the package that we are able to get to make myeloma be a curable disease in the majority. I think we're already curing some, but certainly not as many as we would like.
Right. Well, we are so grateful that you took the time to explain everything for us today. It was an outstanding overview, and as usual, you have a knack for really explaining things in a very easy-to-understand way for patients. So we thank you for that.
Well, my pleasure, and thanks again for having me, and once again, happy holidays to everyone.
Yeah, thank you. Thank you so much, Dr. Orlowski, for joining us today.
All right, bye-bye.
Thanks for listening to Myeloma Crowd Radio. We know that patients can help support the discovery of a cure, and we encourage you to become involved.
Okay, round two. Name something that's not boring.
A laundry? Ooh, a book club. Computer solitaire, huh?
Ah, sorry, we were looking for Chumba Casino. That's right, ChumbaCasino.com has over 100 casino-style games. Join today and play for free for your chance to redeem some serious prizes. ChumbaCasino.com. No purchase necessary. Void where prohibited by law. 18+ terms and conditions apply. See website for details.